Thank you, Penny. So I realised we're keeping videos off because it seems to overdo the capacity. So I've stuck a little picture of me up there so you can see what I look like. I've been working with snails for probably the last 20 years and probably know these snails about as well as anyone would in terms of sort of knowing if they have personalities. A lot of my work has focused on the impacts of stress on learning and memory, but I talk about that quite a lot. So what I'm going to do is look at some other aspects of snails and see if this could be related to them being sentient. So I always like an excuse to pop up a couple of pictures of my dog up, and that's my dog Patrick there. And I think when we're thinking about what is animal sentience, is the ability to feel emotions, happy, sad, pain, fear, pleasure, or the animal's version of those, as has been pointed out very nicely by the previous speakers. In the top picture there, that's my dog doing his agility, and he absolutely loves that. He finds it very exciting. He's very enthusiastic, and you could probably see from the look on his face there that he's really enjoying the experience. He doesn't enjoy quite so much having to pose with his rosettes when he does well, and you can maybe see he's not looking quite so enthusiastic in the lower right picture there. And then the lower left picture, I don't inflict nasty things on my dogs on purpose too much. There he's got snow on his nose and is clearly disliking the feeling of that. And I think when we look at our animals, particularly mammals, and probably particularly our pets, we can definitely sort of view them with the fact that they may be feeling emotions similar to the ones that we feel. When we look at snails, even if, like I've been doing, I've spent 20 years looking at snails, it's really hard to do the same with these sorts of animals. You might think that the one in the middle there is maybe looking a little less happy than the one on the top or the bottom there, but really, how can we tell that? I don't think the snails really express things in the same way, and certainly not in a way that we can understand. So when you look at a snail's face, it's very hard for us to understand that there could be potentially emotions. So why should we care about snail sentience? Well, as already mentioned, if animals are sentient and they can feel experience, they can experience suffering. So feelings like fear, pain, and anxiety, things that we wouldn't want from a moral standpoint, animals to be suffering on our behalf, hopefully not. And we would hope if they do have feelings, that they can also experience joy and happiness. Legislation, as has already been mentioned, also protects in the UK vertebrates and cephalopods, and other species are protected elsewhere, and for the use in scientific research. So the question is, should this also apply to other invertebrates? I think that there are probably quite a lot of people doing research out there whose lives would be made much more difficult if the same sort of legislation applied to invertebrates. And so we maybe need to think quite carefully about whether we do apply it to invertebrates and actually try to come up with some sort of evidence to back it up if we're going to do so. So, can we scientifically determine sentience in an animal like a snail? These are a few ideas I came up with when I was thinking about this question. And to be completely honest with you, whilst I've worked on the impacts of stress on cognition for many years, I haven't really considered sentience beyond keeping my animals happy so that they're behaving in a natural way. And the only thing that really has determined that my snails are unhappy is when they demonstrate maybe extremes of behavior. For example, if they dislike the aquaria they're in and they're stressed by the environment, they'll crawl out of the water and literally commit suicide. Or I may see high mortality if they're kept in poor conditions. But luckily, over years of experience, I've learned how to keep snails in conditions they generally don't seem to exhibit these extremes of. But can we look at other aspects of the behavior to determine if they may be sentient, and therefore if we can assess their happiness or their suffering in a more precise way or a better way, so that we can improve conditions for them. So the list I've come up with there is by no means a ubiquitous list, and there are other things that have been mentioned already today that could also be possible to look at in snails, but I'm going to look over these 4 things. So we have nociception versus pain. So what is the response to these noxious stimuli, whether they avoid punishment or try to acquire reward, potentially through learning and memory. And as I mentioned, I've worked quite a bit on the effects of stress on memory, but I'm not going to go on into that today. Do snails demonstrate any level of self-awareness, and do they demonstrate cognitive bias? So can they act in a pessimistic or optimistic way? So if we start off first of all with the nociception versus pain. Nociception, sort of the immediate withdrawal from a noxious stimulus, whereas pain is typically indicated by prolonged changes in behavioural physiology from that noxious stimuli. And it's been demonstrated that anaesthetics and analgesics have shown reduced responses to noxious stimuli in snails. So for example, sepia on the top there, the garden snail, if you apply opioids to the animal or bathe it in opioids, it increases the amount of time it takes to withdraw from a heat stimulus. So they have a higher tolerance for a heat stimulus that could otherwise be damaging. And those people who work with snails quite often use ethanol or methanol, for example, to prevent snails withdrawing into their shell to be able to carry out surgical procedures on the snails. And whilst some of the anaesthetics, analgesics have been looked at in terms and how they impact on the animals have been looked at in terms of their survival and sort of some basic behaviours immediately post-surgery. So do they become active again fairly quickly? There's been very little done or nothing to my knowledge done in terms of evidence for longer term changes in behaviour or physiology. So if we apply a painful stimulus to the snail, or what could be a painful stimulus to the snail, in the absence of anaesthetics or analgesics, does it result in a longer term behavioural or physiological change than if we apply that same stimulus in the presence of anaesthetics or analgesics? That hasn't been done, but I think it is something to look at in the future. And what it does do is provide opportunities to potentially test whether snails feel pain using some of the avoidance training paradigms that we've used in research with them. So one of the things that we do see with snails, and we have got good evidence for, is that they will perform behaviours to avoid punishment, to avoid noxious or negative stimuli. One example of this is conditioned taste aversion. In this case, the snails are food deprived for 24 hours prior to conditioning, so we're increasing their desire to respond to food stimuli. If you apply a sucrose solution to the snail, and here we have a snail in a petri dish above a mirror, and hopefully you can see there the mouth is opening. So they perform this biting behaviour on the surface whenever they sense food in their environment, which includes sucrose or vegetable extracts. And they can be trained to associate the presence of sucrose solution, if they haven't experienced it before, with a negative experience. So, for example, exposure to potassium chloride, which potassium chloride generally causes the snails to withdraw from that stimulus, so they move back into their shells. And we can measure how good their memory is of this by measuring how much their bite response to sucrose changes following this negative association and compare it with controls. So here we have an example of conditioned taste aversion. We have naive animals and how they respond to sucrose. Indicating on the wrong slides there. So naive animals and how they respond to sucrose with about 12 bites per 20 seconds. Those individuals that have been backward conditioned, and that's where you expose them to potassium chloride and then sucrose, so that the potassium chloride is not, so that the snails are not expecting the nasty negative stimulus to come after sucrose, and then those that have been conditioned for taste aversion. And hopefully you can see here on this figure that those that have undergone this conditioning paradigm where the sucrose has been applied and then potassium chloride reduce their bite response to sucrose. And we see very similar responses if we use other negative stimuli. So if you pair sucrose with an electric shock, or if you pair sucrose with a strong physical stimulus, both of those will reduce the bite response. So in this case, a noxious stimulus is having priority over other stimuli. For these snails that have been food deprived for a period of time, the snail is no longer responding appropriately to a potential food resource. They're making a decision that the feeding behavior in the presence of food, they're not going to perform that behavior because they're predicting potentially the appearance of these negative stimuli. Another example of this is using operant conditioning of aerial respiration here. If you place the snails in a hypoxic environment, they come up to the surface of the water and they breathe using this orifice here called the pneumostome. So it basically opens to a basic lung. And if you place them in a hypoxic environment and every time they attempt to do this, you use a wooden stick to prod them on the pneumostome, then they quickly learn to stop trying to perform this behavior. And what we see is a decrease in the amount of time that they try to perform aerial respiration. So again, we have this aversive stimulus, this poke that they're getting, this physical stimulus is taking priority over the stimulus of the hypoxic environment, which is reducing oxygen availability. And so they're no longer performing this behavior that's appropriate to gain oxygen. But we also see that snails can change their behavior for acquisition of a reward. We can use appetitive conditioning. In this case, snails can be trained to associate a novel odor, amyl acetate, which smells a bit like pear drops, or gamma-nonylactone, which smells a bit like coconut, with the presence of food. The snails don't respond to these chemicals until they have formed an association. But if we train them by giving them 2 minutes exposure to either one of these chemicals and then 2 minutes exposure to sucrose, we can then train them that that smell is associated with a food availability. And those individuals that have received contingent training show a significant increase in the bite response relative to those control individuals that have not received the association when they're exposed to those odors alone. So in this case, the novel odor is associated with the presence of food, resulting in an increase in feeding behavior or an increase in the acquisition of the reward. Another thing that we may be able to do with snails is whether we can determine self-awareness, or a question, can we determine self-awareness in snails? One of the tests that's often used in a lot of species to determine if animals are self-aware is the mirror test, where the response of the animal to its own reflection in the mirror is compared with the response to an unknown conspecific. If these responses differ, it's thought to indicate that the individual who's viewing themselves in the mirror is able to recognize that individual as self. But snails, unlike the cephalopods that we heard about in the last talk, have pretty poor vision. Their visual acuity would not be sufficient, I don't think, to recognize one snail from another, though they may be able to recognize the shape of a snail. So it's unlikely their visual acuity is sufficient to do that. But snails live in a chemosensory world, and we have a lot of evidence based on chemical cues that snails are able to identify self. And a lot of this comes from trail following. Snails use trail following in a wide range of different purposes. They lay down these mucus trails and they can use these to identify self and other individuals. For example, if we're looking at mate search behaviour, if you isolate a snail and then you place it into a behavioural arena with its own slime trails, it will typically follow this sort of pattern, where it will search around the arena, sort of performing circular movements. But when it hits its own trail, it won't change direction. It will just continue straight across it. If you isolate the snails and then place them in a behavioural chamber with a conspecific trail in there, they will, when they hit the trail, frequently follow that trail in the direction that the conspecific snail was moving. So around 25% of the time in this species, but it can go much higher. So in that case, when they're mate searching, they're avoiding self-laid trails, but purposefully following conspecific trails. Conversely, if you look at homing gastropods, what you find is that they ignore conspecific trails, so typically show negative responses when they come across a conspecific trail and don't follow it, versus a positive response to their own trail. So if they come across their own trail, they will orientate around the trail, work out the direction of travel, and if they're going back to their home, they'll move in the opposite direction of travel to the way in which they came out on the foraging trip. So we've got two clear examples there of the fact that snails will differentiate between conspecific trails and self-laid trails. So they are able to identify their own chemical cues. Another aspect that we could look at in snails is cognitive bias. And cognitive bias has been looked at quite successfully in bees, for example, if we're looking at, for examples in invertebrates, and also, I believe, in cephalopods. To test cognitive bias, we need to train the animals with two different stimuli. One of those stimuli we associate with a negative experience, so indicated here in blue. And then the other stimuli we train to associate with a positive experience, indicated here in yellow. When we come to test them, we can then test using ambiguous stimuli and see how the animals respond. Do they respond to the ambiguous stimuli as if they're a positive stimulus or a negative stimulus? And how does their experience impact on that response? We've done a little bit of work with snails, not got quite as far as we would have liked, but here we've come back to those novel odours, the amyl acetate and the gamma-nonylactone, so pear drops and coconut, and trained the snails to associate one or other of those novel odours with a positive stimulus, so with the presence of sucrose, and the alternative odour with a negative stimulus, the presence of potassium chloride. And you can train them over several days. We've only run it for a couple of days so far, but the snails receive either the positive or negative training in different orders. And we switch round whether we use amyl acetate as the odour associated with the positive stimuli or the negative stimuli. So here, for example, at the top, they received a positive training stimulus, so one of the odours paired with sucrose, followed by a negative training stimulus, one of the odours paired with potassium chloride, and then the same order on day 2. And we switched around what they received on each day. And then we tested the response of the snails to a 50-50 mix of odours. So we've given each snail 2 experiences of the positive association and 2 experiences of the negative association with the different odours. How do they then respond to a 50-50 mix of those odours, giving them this conflicting information? Well, what we found is that snails appear to prioritise their most recent experience. So here, if they received the positive association stimuli, so the odour paired with sucrose second on their final day of training, they showed a much higher bite response than those individuals that received the negative stimulus last. But it's worth noting that this response here, this bite response of 2 to 3 bites per minute, is higher than we see if we just carry out negative training. So if we carry out negative association training on its own, we generally see 0 biting behavior. So generally, it appears that snails are optimists and they expect to get some sort of food resource rather than the potassium chloride when they're presented with this ambiguous cue, this mix of the different odours associated with the presentation of sucrose and the presentation of potassium chloride. They go on they sort of veer towards the optimistic side that food might be coming. I think the next step we're going to look at with this is whether we can manipulate their positivity using additional negative or positive experiences. So, for example, if we expose them to predator cues, would that err them towards the side of being more negative and showing lower levels of bite response? So if we come back to the list that I had at the beginning, can we scientifically determine sentience in a snail? Well, at the moment, we don't have really, I don't think, strong evidence for nociception versus pain. Yes, they show responses to what are potentially painful stimuli that analgesics or anesthetics prevent, but we haven't looked to see if there are any sort of longer term changes in behavior or physiology. Avoidance of punishment and acquisition of reward. I think we have clear evidence that snails have the cognitive ability to avoid negative stimuli and respond positively to associations that gain reward. And we have a lot of data supporting that. So I think we can definitely tick that box. When it comes to self-awareness, I think we have clear evidence of self-recognition, but is this really self-awareness? And if we come back to the dog here, we've got a nice blow-up of the nose. There was a paper by Horowitz who showed that whilst dogs don't pass the mirror test, they are quite capable of identifying their own urine versus other dogs' urine. So maybe we need to start thinking more in a chemosensory world for a lot of animals when it comes to their ability to self-recognize. And cognitive bias, pessimism or optimism. I think snails, based on the experiment we ran, seem to be generally quite optimistic, but we want to look at whether we can manipulate that pessimism and optimism, whether we can create more optimistic or pessimistic individuals based on their experiences around that training. So in conclusion, we have some of the tools I think we need to test traits which are considered to allow the recognition of sentience in animals. But I think we need to do some more work with the snails to really get some good evidence if we want to argue that the snails are sentient. And I think one of the things that snails have definitely taught me is that we need to carry out research in ways that are relevant for the species. And for aquatic snails, this is largely carried out in a chemosensory world rather than thinking about a visual world. Questions or comments, welcome.
Great, thank you very much indeed. I'll just say, before we move on to questions for you, Sarah, I was just wondering whether people would rather skip the breakout groups and have a bit more discussion with you and the other speakers. What do people think? Do you want to put yes in the chat box if you want to do that, or no if you want the breakout groups? Yeah, I thought so. Okay, so let's have a couple of questions just for Sarah, and then we can, yeah, okay, I got the message. That's a resounding yes. So, because I just noticed there was an interesting question from Kirsten Clappert that I think we could put to all of the speakers. And then there's a question from Jonathan and also a question from Phil Brooks. So perhaps Sarah, if you'd like to take those two questions and then we can open it up a bit.
So from Phil and Jonathan, yeah.
Yeah, you can see those, yeah.
So Phil, I used copper tape to keep snails out of my veg patch. One approached, first tentacle retracted, immediately it hit the copper. Snail persevered, second tentacle retracted instantly on touching the copper, and snail changed direction. Notably required 2 noxious stimuli, rather like the decapods requiring 2 electric shocks to learn avoidance. So would be interested in, where are we? Sorry, every time somebody types it pops out, pops away. Would be interested in knowing if this resulted in long-term avoidance of the tape. So I guess that's something you could probably run in a small aquaria to see if they will avoid an area where they've come across copper tape. Long-term memory in snails is generally around 6 hours plus. So I would say if the snail didn't come back that day, it's demonstrated the neurophysiological changes that we would expect associated with the long-term memory formation, but long-term memory formation can last for different lengths of time. But I think that would be quite an easy experiment to run to see if you could get some place avoidance. So I would say, thinking about my aquatic snails and probably related to terrestrial snails as well, you might want to give them some sort of odor cue if you were running similar sorts of experiments. I imagine that they were attracted to your plants that you were trying to keep them away from by odor cues from the vegetables.
Thanks. And Jonathan was asking whether there'd been any work in snails like Bob Elwood's motivational trade-off work in hermit crabs.
Yeah, I mean, I guess that when we're running a lot of the learning and memory work, we are giving them motivation to behave in a particular way. So in the food example, we're giving them motivation to feed by food depriving them for a short period of 24 hours. When we're carrying out the operant conditioning, we're giving them motivation to carry out aerial respiration by exposing them to hypoxia. Neither of those exposures is particularly detrimental to the snails. They would frequently go for periods of time without feeding in the wild, and they live in very severely hypoxic environments. I know somebody in one of the labs kept them in hypoxic water for about 48 hours and they fully recovered from that. So the motivation we're giving them in terms of the behaviors that we're training them not to do isn't necessarily particularly strong, but could be manipulated fairly easily, for example, for starving them or food depriving them for longer periods of time, or placing them and holding them in a hypoxic environment for longer periods of time. But I guess those are fairly negative things to do to snails. So balance up of whether increasing the motivation to do these behaviors versus the stimuli that they're trained with. There has been some work looking at how strong stressors are and how that relates to the impact of stress on memory. So thinking of the sort of the Yerkes-Dobson curve, where you have sort of low levels of memory at very low levels of oh, my brain's gone. At low levels of stimulation, but then you have the sort of mid-level stresses can actually be quite good for your memory, and then you go down and then you go out the other end where you have very high levels of stress, which can then prevent memory. So there has been work looking at that.
Great, thank you. And I think we've got a few more questions coming in. I'd like to ask you sort of a combination of a couple of them, and then maybe we could move to some of the more general questions. So there are a couple of people have asked you what the implications are. So what sort of refinements would you suggest for snails in the lab, and how should humans change their behavior relating to snails, like using a torch in dark and wet alleys, avoiding lethal pest controls, being gentle in the garden? What do you feel is proportionate? So what at the moment, given the current understanding of snail sentience or not, what precautions do you feel are proportionate in the way we interact with snails?
I mean, I think, you know, Jonathan sort of highlighted the fact that we should use the precautionary principle, and if there is a possibility that they may be sentient, then we can follow the best guidelines that we have in place in terms of things like euthanasia, for example. Sprinkling salt all over your snails probably isn't a particularly nice way to dispatch them. And I do avoid trying it, but whereas treading on them is probably, if you're destroying the central nervous system, or is probably or central ring ganglia, I should say, for the snails, is probably a much nicer way of dispatching them, much faster if you can do it very effectively. I mean, I certainly try where possible to keep my snails happy in the lab, and as I said, it's very hard to determine beyond their behaviour, but I've certainly stopped the crawl out behaviour that I've had previously, where snails, baby snails in particular, occasionally committed suicide by crawling out of the water, and I've managed to keep them a lot happier from that perspective. And occasionally they seem to like the bubblers. You see them going, you see occasionally the snails sort of going around in circles and going up in the bubbles. I'd like to think they were playing with the bubbles, but
well, they're certainly voting with their foot, aren't they, if you're managing to keep them in the tank? Yeah. So that sounds positive.
And they do seem to not like being on their own as well. I know that there's been work looking at how it affects their mating behavior, and so there'll be direct physiological effects on them through their inability to mate if they're kept in isolation. But I've also seen what I think are negative impacts on their general well-being, keeping them in isolation as well for periods of time. Right. So don't isolate your snails.
Well, you've certainly made me think about them differently, and I had quite an open mind to start with. So thank you very much for all that. That was fascinating. I'd just like to go back, scroll up to Kirsten's question, because I think this is highly relevant for all of our speakers, and I think it's a good point. So Kirsten says, thank you very much for all these very interesting talks, but I do start to wonder if being able to learn associations, which is under natural selection and probably genetically hardwired in basically all animals, can be used to think an animal is sentient. Also, pain responses are a survival guarantee, just wondering. So would any of the speakers so far like to come in on that one?
I'm happy to do so if you want.
Okay.
Yeah, I mean, I mentioned in my talk that it's a subject of ongoing dispute which kinds of associative learning are evidence of sentience and which are not. In that you're right that basic kinds of associative learning, are present in snails and are present in sea slugs, Psychoplesia, and that's been known for a very long time, and are present in nematodes as well. So it would be too quick, I think, to assume that any associative learning straight away already means you've got enough evidence of sentience. I mean, 2 points about that. Firstly, I think we have to look to the human evidence to try and discern which sorts of learning are facilitated by subjective experience and which are not. I've written about that in a couple of papers that I can post in the chat. But also I think that when we're thinking in a precautionary way about sentience, and we're thinking about our policies at a local level and about our animal welfare laws, it's reasonable in the face of that ongoing scientific dispute to see any associative learning at all as a potential warning sign. And one of those 8 warning signs that I listed in the talk. So that's how I see it.
Thank you. Would anybody else like to come in on that one? Any other thoughts? People working in the different species groups.
I just wanted to make a short comment. This is Jennifer. I think one ought to treat one's research animals and the wild animals around one and the domestic animals that we are associated with. We ought to treat them with care anyway, regardless of whether we have decided they have sentience or potentially sentience.
Yes, that's a good point. That's a valuable point, and it's certainly one that my organisation embraces. I guess it's just the issue if you're trying to influence legislation or codes of practice, and then you do have more of a demand for evidence, and sometimes it can be quite tricky when you're sort of politically, thinking particularly about some of the points that Helen raised earlier and that Sarah's just been talking about in relation to snails. It can be quite a fine line between bringing the public and legislators with you and people feeling that it's just that you'll get arrested for squashing a fly and that what you're calling for is completely ridiculous. That's where the evidence becomes so important, I think.
I think evidence genuinely is important, not just for strategic or political reasons, but because without evidence, we don't have any idea what to do in order to protect the interests of animals. And we could easily find ourselves doing something that we think is, more humane. But if it's not evidence-based, it could easily be less humane.
This is Catherine talking later on reptiles. I'd like to echo that point from Jonathan, specifically for pain, because if we're thinking about that nociception and pain both evolved under natural selection, but if we want to do something practical for the animals concerned, it may be that in some animals where we experimentally believe there's nociception but not pain, local anesthetics, sodium channel blockers may be more effective. Whereas if we have evidence of central pain processing and the action of opiates, for example, that can be something practically that we can add on. So it does have practical implications, whether how we discuss nociception and pain and how we use that knowledge going forward kind of practically as well.
Yes, thank you. We've got a question in the chat box from Lynn. Can everybody see that? Who'd like to come in on that one?
I'm going to confess, sorry, it's Sarah here, to squashing quite a few mosquitoes in my time. But then I think they're also very ecologically important animals. They're food for an awful lot of species. So, yeah, I think on balance, it's not just about us, it's about the environment as well and the other species that rely on them.
I think so. That this, sorry, somebody else going to say something. Go ahead.
It's Helen here. I was just going to say it's an interesting question, because if you think not just about disease but about danger, so if you're facing a tiger, versus a mosquito, you're more happy to squash that mosquito and that malaria-carrying mosquito versus shooting a man-eating tiger. It's really tricky because I've squashed plenty of mosquitoes in my jungle days, that's for sure. But if they're sentient, I may feel a bit different if I knew for sure. Tricky one.
There is one more simple or less complex principle, though, isn't there? That you don't cause animals avoidable or unnecessary harm, which I think also applies across the board, and that enables you to apply a sort of a harm-benefit assessment when you're thinking about doing anything negative to any animal. So it's justifiable to kill or squash a malaria mosquito if they've got the potential to infect a human and cause human suffering. And if you're thinking about, say you have unwanted rodents in your house, then the onus is on you to make sure that you're disposing of food responsibly and blocking up holes, etc., and trying to deter them before you go on to go straight to an exterminator company and get them to poison them to death. So I think in each case, you look at whether it's justifiable to do something to harm that animal, and if it is, how you can avoid and mitigate those harms. So that's a sort of an easily defined universal principle. But I do feel that we all are inherently speciesist and sizeist, and it's really important to question and challenge that. Axelson says, yes, you do kill huge numbers of animals every time you take a walk in the forest. That's true. It's impossible to live on this planet without disadvantaging and directly or indirectly killing other animals. But it's just about understanding and recognizing that sentient animals have an intrinsic worth, perhaps, and that we need to think about how we can minimize impacts on them.
I'm sorry, just going to say, to some extent, we have to stop this anthropocentrism, because we're doing incredible damage to the planet just by pretending that it is only our worth, it is only our thriving, if you like, that is necessary. And so in the process, we're killing whole species just because they're inconvenient to us.
Yes.
I was going to say it's interesting to look at the literature, because I have a paper in the journal Animals about how we ignore invertebrates. And one of the things I found when I looked at research on interaction with insects in general, that it's all about how to kill them.
Yes. Yes. Could we could you put the link in the tab to your paper, or I could find it instead. I see Jonathan's also put a link to the paper in the chat box.
I'm afraid I don't know how to do that.
Okay, I'll have a look. It was in animals. So it was in animals.
The journal's called Animals, and it was the year before last, I think.
Okay.
I published a paper about the amount of attention that we extend to invertebrates. And I should also mention, by the way, that Claudio Carrere and I have recently published a book on, hang on a minute. Okay, it's the Springer series on animal welfare, and this is called The Welfare of Invertebrate Animals. Carrere and May, their editors.
Right, thank you. Oh, sorry, I've just tried to paste the animals paper in the chat box, and it's gone wrong. I don't think that's going to work for anybody. I'll have another go at that in a minute.
Well, it was interesting. We went out and found specialists in spiders, in bees, in crustaceans, in cephalopods. We really tried to look at a wide variety of the invertebrates. And I remember when Claudio first was contacted by the Springer people, and he got in touch with me and said, can we do this on the welfare of invertebrates? And I said, oh, the other 99% of animals on the planet.
Yes, exactly. Oh, we have right. So there's a question from Phil about bivalves, but I think if we drop down to Eleonora, we'll come back to Phil's question. What about eradicating alien sentient species which are invasive and damaging other animals, e.g. the ring-necked parakeets invading Europe? Would anybody like to comment on that?
I'm happy to. I mean, yeah, that issue of where welfare goals come into conflict with conservation goals is a fascinating area on which it would be great to see more work. I mean, sometimes the welfare goals and the conservation goals align, and that could be the case with the parakeets. I mean, if they're doing a lot of harm to native species by displacing them from their nesting sites, there could end up being a welfare case for taking action. But I agree that in some cases there is genuine conflict between welfare goals and conservation goals, and no agreement really on the right process for resolving that conflict, which is an interesting problem.
There used to be the compassionate conservation movement. I think, I don't know if it's doing masses at the moment, but that was really to bring together conservation and welfare to sort of more aligned goals, because you're right, the two conflict all the time, but there is a potential to actually have some more synergy there, because these are alien invasive species is generally usually human caused, so we're at fault, and the parakeets shouldn't be the ones to suffer because of that. But sometimes decisions have to be made. But yeah, more synergy is definitely needed there.
Well, to some extent, we can say it is a normal survival of the fittest. Granted that we were the ones that intervened to set up the situation, but do we have the right to, in this particular case, play God and decide that we don't like one animal being where it is?
Yes, I think that there's just thinking particularly about all the discussions at the moment to use genome editing and gene drives in order to control unwanted populations of animals, including mammals. There's ongoing work now to look at gene drives to reduce numbers of grey squirrels and mice that are harming island populations. And it's important I think where animal sentience can help with this, or where understanding animal sentience can help with this, is in reducing the tendency to take a very sort of anthropocentric approach to this, and to understand that all of the animals involved are capable of experiencing suffering, and it's not any one of those individual animals' faults. You know, they're just naturally exploiting a situation that's being created by humans. And I see these kinds of considerations coming increasingly into the debate as to how to reduce numbers of unwanted wild animals, whereas previously it would just be a case of attempt to eradicate them at all costs. I think decision making is becoming more measured now, and there's more of a drive to consult publics and see how they feel about it, which can only be a good thing. And again, I think sentient science can help to inform that too. Should we just come? It's nearly time to break now. Should we just come back to Phil's question? So it's going back to the mollusks again. Phil's asking, bivalves have 3 pairs of ganglia controlling different parts of the animal and processes. Snails have a central nervous system, but is there central processing? Does this make sentiency more likely, or might it be possible in an entirely decentralized nervous system? So would either Sarah or Jennifer like to comment on that one?
So I'm sorry, Sarah here. I don't know that having a central processing is necessarily necessary for sentiency. It depends on what we decide or what is judged to be sentient. So if experiencing pain, I think, could be potentially very possible in a disparate nervous system, where you have different ganglia controlling different regions of the animal, doesn't mean that a particular region couldn't be experiencing pain, even if that isn't processed throughout the whole animal. Could be one viewpoint. Certainly the snails within the central ring ganglia do use information from disparate parts of the animals and process that in a way. So if you look at, for example, their response to stressors or environmental stress, that comes through a variety of different sensory neurons, but can influence neurons where you, or the activity in neurons where you see memory of these different behaviors. So there's definitely some sort of central processing going on of the information.
I've been thinking about that. I think that sentience is more likely to be the result of a complex nervous system. And of course, complexity could be in many different loci. But I think that the tendency for evolutionarily for animals to gather the processing in one area, which we call a brain, I think it makes it more likely. It doesn't mean it couldn't happen in a distributed nervous system, but I think it's more likely if there's a central area with more complexity and more interneurons and more decision-making areas.
Thank you.
I think
sorry, go on.
Sorry, I think one thing we need to bear in mind is sort of the replication of function that you get in a lot of brains that isn't necessarily the case in mollusks. So they may have a lot fewer neurons, but individual neurons are doing specific jobs, whereas you may have replication of that function in what appear to be more complex brains with more neurons. Doesn't necessarily mean greater function always.
Yes, the insects are a good example.
Yeah. Okay, thank you very much. I've just before we break, I've just put another poll up there, which some of you are starting to respond to. If you could click on the little poll icon, I would just be interested to know before we break whether anyone's changed their mind regarding which animals are sentient. So most people are saying no. A couple of people now believe that fewer animals are sentient. Most people are saying, well, most people are saying no, but second to that, now I believe more animals are sentient, about 30% of you. So that's quite an interesting result. I was just wondering how people would feel. Thank you for your votes. So it's now past. We've got an hour's break scheduled in for you now to do whatever it's appropriate to do at this time of day, wherever you are. Thank you very much for your questions, and thank you very much indeed to all of the speakers. That the talks have all been brilliant and fascinating, and certainly given me a lot of food for thought, and they've obviously given you a lot of food for thought too.